Welcome back to BinaryBio. If you're interested in bioinformatics and problem solving, you're in the right place. Today, we are diving into our Rosalind problem series to tackle a fundamental challenge finding a spliced motif. But first, why does this matter? In biology, DNA isn't just a solid block of instructions, it's full of filler and functional parts. A motif is like a high priority bookmark. These short sequences often tell a cell where to start reading a gene or where a protein should attach to turn a gene on or off. By finding these patterns, we can predict how a cell will behave or even identify how different species are related. So, what is a spliced motif? Imagine trying to find a secret word hidden inside a massive book. The letters of the word aren't right next to each other, they are scattered across different pages. But, and this is key, they still appear in the correct order. In code, we call this a subsequence. Let's look at this Rosalind problem. We have two DNA strings. String S is our long main sequence, and string T is the short motif we are hunting. We need to find the locations in the big string where G, T, and A appear in that specific order. First, we find the G. It appears at index 3. Next, we need a T, but it must be after that G. We find it at index 8. Finally, we need the A appearing after the T. There it is at index 10. Our output is 3, 8, and 10. Note that Rosalind uses 1 based indexing, so we count starting from 1, not 0. The problem says we only need to find one valid solution. This allows us to use a greedy approach with a two-pointer technique. We'll use one pointer, motif pointer, to track the letter we are currently hunting for in our target motif. We use a second pointer, S pointer, to scan through the big DNA string. The logic is simple. We move through the big string one letter at a time. If the letter we land on matches what we need, we save the location and move our motif pointer to the next letter in our target word. We never look back, which makes this algorithm incredibly fast. In Python, we initialize both pointers at 0 and create a list called res for our results. We use a while loop that runs as long as we haven't reached the end of either string. Inside the loop, we check, does the current DNA letter match our target letter? If it does, we move the motif pointer forward and save the current index, plus 1 for that one based indexing. Regardless of whether we found a match, we always move S pointer forward. This ensures we keep scanning through the DNA in the correct order. When we run this on our sample data, we get our result. Since this algorithm requires only a single pass through the DNA sequence, it operates in linear time. While the technical complexity is O of n plus m, we simplify this to O of n because the genomic sequence is the dominant factor in the computation. In a lab, scientists often work with cDNA, which is a clean copy of a gene with all the junk removed. To understand where this gene came from, we have to map that short cDNA back to a massive genome. This is exactly what the spliced motif problem represents. The cDNA is our motif, and the genome is our search string. Because of introns, the cDNA won't match the genome in one solid block. It matches as a subsequence. By finding these positions, we are performing exon mapping, identifying the exact coordinates of the functional parts of our DNA. We also use this to compare different species, like a human and a mouse. This is called synteny analysis. Even though species change over millions of years, the order of important genes often stays the same, even if the distance between them grows. If we can find a motif from a human as a subsequence in a mouse, it's strong evidence that they share a common ancestor. This helps us identify orthologs, which are genes that perform the same function across different branches of life. If you found this helpful, hit the like button and subscribe to BinaryBio. In upcoming videos, we'll solve more Rosalind problems and gradually build strong bioinformatics problem solving skills. Thanks for watching. See you in the next one.